for tuning in. Welcome to our segment on the Floating Disk Lab.
As I head to the lab now, it is imperative to understand that photosynthesis provides the Earth with oxygen, and the rate that photosynthesis occurs can be measured by how much oxygen it gives off and how much carbon dioxide is consumed. Normally, leaves or leaf disks float because they have carbon dioxide and oxygen in their systems. However, if gases are removed, the leaves will sink and photosynthesis will not be able to occur. Photosynthesis can occur if there is an alternate form of carbon dioxide, in this case the sodium bicarbonate, which oxygen and sodium bicarbonate are exchanged to the stoma and the leaf becomes buoyant. This lab is an indirect measure of photosynthesis.
In order for this lab to be complete, one must know the equation for photosynthesis. The equation is the combination of water, carbon dioxide, and light energy to produce glucose and oxygen. Now to the fun part! Materials necessary to complete this lab are as followed. Baking soda, which will be used as your sodium bicarbonate, liquid soap, 2 plastic syringes, living leaves, a hole puncher, 2 clear plastic cups, a timer, and a light source. We predicted and hypothesized that the leaf discs placed in the sodium bicarbonate would react and would float faster and at a higher rate than those leaf discs that were placed in the distilled water. Within this experiment, the independent variable is the type of solution, water and water with sodium bicarbonate, in which the leaf discs are placed in. The dependent variable is the time it takes for the discs to float. Every minute data is recorded. The control was the water solution without sodium bicarbonate. The constants are the amount of leaf discs. Only one trial was conducted.
According to the data, every minute for 20 minutes, data was recorded. It appears that one of our leaf discs placed in the cup with only water floated to the top. This was because the leaves did not have a source of carbon dioxide that they could use to perform photosynthesis. Meanwhile, the sodium bicarbonate disc did not start to float until 5 minutes. By the end of 10 minutes, 5 discs had floated to the top. After 15 minutes, 7 discs had floated to the top in the sodium bicarbonate cup. At the end of 20 minutes and/or experiment, 9 discs ended up floating sodium bicarbonate. The results supported our prediction that discs in the sodium bicarbonate would float faster and at a higher rate than those in distilled water. Within this graph, one can see that the distilled water discs remain constant. Only 1 leaf disc floats, meanwhile 9 sink in the distilled water. This is represented by the blue line on the graph. Meanwhile, the red line resembles the number of discs that floated in the sodium bicarbonate. At minutes 1, 2, 3, and 4, only 1 leaf disc floated. It wasn't until the 5th minute that the amount of leaf discs floating increased. The mode, or most occurring within the data, would be 7 discs floating. All in all, the distilled water remained the same, constant. Meanwhile, the sodium bicarbonate infused discs increased exponentially, as seen in the graph above. The first group we compared data with was a group that utilized a green light versus the incandescent lamp, which we utilized. The green light caused 0 chads to float within the distilled water. Meanwhile, the incandescent light allowed for 1 chad to float in our distilled water solution. Also, this group found that in the green light, only 1 chad floated in the sodium bicarbonate versus 9 of ours that floated by the end of our experiment. The chlorophyll of the leaf discs do not absorb green light as quickly, therefore photosynthesis cannot occur. Meanwhile, our incandescent light is made up of blue, red, and green light, which allows the plant to reflect, giving the chad its color and absorbing other 2 colors for energy. Meaning, chlorophyll absorbs red and blue light more readily than the green light. We were the control group and therefore made no changes to the instructions and/or experiment. Group 1's experiment remained constant with CO2 and without CO2 and the green light. Meanwhile, ours expanded exponentially, especially in the sodium bicarbonate solution. The second group we compared data with decided to add an extra 1/8 teaspoon of sodium bicarbonate to their solution. In doing this group developed and discovered that by adding a 1/8 teaspoon of sodium carbonate, all 10 chads floated at the 10-minute mark. These chads remained floating for the duration of 10 minutes. This occurred because there was more carbon dioxide available for photosynthesis. Meanwhile, our data, aka the control, never had 10 discs float. I believe we did not have 10 discs float like this group did because we did not have extra or enough carbon dioxide to do so. The leaf discs absorb carbon dioxide from the atmosphere and give off water vapor and O2 all through the same openings, the stomata and leaves. The issue is that if there is not enough carbon dioxide in the environment, the stomata would be open for too long and would lose too much water vapor and dry out. The third group we decided to compare data with used ice water in order to determine how many discs would float. The ice water caused 0 leaf discs to float within 20 minutes. The ice water did not allow for a sufficient amount of light to pass, and the cold water caused the enzymes to move at a slow rate. This slow rate results in less discs making it fully through the photosynthesis process and floating. As the temperature of water decreases, so does the rate of the reaction. This is the reasoning behind 0 discs floating within 20 minutes. Meanwhile, we had 9 discs float, this group had 0. If the temperature was room temperature or warmer, more chads for this group would have floated. All in all, it is clear to see that out of all the controls that were tested, that the best results occurred with the incandescent light and when a 1/8 teaspoon or more of sodium bicarbonate was added. Adding more sodium bicarbonate resulted in all 10 chads floating, for more carbon dioxide was present for photosynthesis. The ice water decreased the temperature below the enzyme's optimal temperature and resulted in 0 chads floating in 20 minutes. When temperature decreases, so does the rate of enzymes and photosynthesis. Within this lab, it has been concluded that a solution with more carbon dioxide will have the highest rates of photosynthesis. When there is more carbon present, more carbon can be fixed, therefore making photosynthesis quicker and easier. The distilled water solution took the longest for photosynthesis to occur, because there was not enough carbon dioxide in the water, making photosynthesis occur slowly. One may ask, what was the purpose of creating a vacuum with the syringe? Well, the leaf discs normally will naturally float. However, the vacuum causes the air spaces in the leaf discs to be filled because it takes the air out of the leaves. As water fills the leaves, the density increases and the leaf sinks in the syringe. We also remove the air from the leaf chad's central vacuole and was being replaced with water. For this procedure, we wanted the discs to sink because once placed under the light and in the bicarbonate solution, we can tell the process of photosynthesis is taking place as the discs rise. Another question one may ask is, where did the leaf discs get the carbon dioxide needed for photosynthesis? The sodium bicarbonate ion acts as a carbon source for photosynthesis, making the discs sink. These discs end up removing carbon dioxide from the baking soda that was mixed with the distilled water. This was the start for the photosynthesis. What made the disc leaves float? As the process of photosynthesis continues, the leaf converts the carbon source into oxygen. This oxygen is then released into the interior of the leaf, causing the buoyancy of the disc to change, which makes the leaf float. When the leaf discs were put into the distilled water and the sodium carbonate, carbon dioxide re-entered the chad, which causes it to float.
In every experiment, there are always sources of error. In our experiment, we think that the pipettes lost water within the process of creating a vacuum for the leaves. Therefore, there may not have been exactly 2 to 3 centimeters like the experiment asked for. Also, when we stirred the solutions using the pipettes, we did not take into account the CO2 pipette mixing with the distilled water. These errors could have created different results.
An error in comparison 1 group data was the incandescent light that was near their green light. The light from the incandescent light from another group could have also reached their cups. According to them, their CO2 cup was closer to another group's incandescent light, which could have skewed results. An error in comparison group 2's data was that they did not stir the cup to get the discs off of the sides of the cups until a few minutes in. An error in comparison 3's data was that they applied the head shields too late, which fluctuated the temperature. All in all, by completing this lab, we learned the importance of having all reactants of photosynthesis. Since the distilled water lacked carbon dioxide, less leaf discs fully photosynthesized. We also learned that environmental variables like light intensity, color of the light, temperature, and pH all affect the rate at which photosynthesis occurs. We also discovered that sodium bicarbonate increases the rate of photosynthesis by providing the reaction with extra carbon dioxide. The produced oxygen in this case was the byproduct. The sodium bicarbonate ions act as a carbon source and cause the leaf discs to float at a faster rate. The ice water or a decrease in temperature slows down enzymes and decreases the rate of photosynthesis with it. A warmer temperature increases photosynthesis reaction time.